Hello, students. This is the lecture for Chapter 10, Part A, and the focus of today's lecture is on Mendelian genetics and the inheritance of traits. As usual, I have included in the lecture three slides that you can use for note-taking. Now, if you are thinking that you're going to be having some problems with printing, then as we had talked about the other day, please write it down in your notebook. And then what I want you to make sure is to put a section heading or record the questions that are pre-printed on these lecture note sheets and then answer them such that when you upload the work, I could find your answers quickly. All right, so let's begin. So how does the inheritance of dominant or recessive alleles affect phenotypic expression? One of the activities I had given you last week before we had this official start of the week was that I wanted you guys to take some surveys of your own, both about yourself and also on your family members, to find out what kind of traits do you and your family members have that represent dominant or recessive traits. And thanks to all of you, I have gotten your results, your individual results, as well as your families. And because you took part in that, we have been able to significantly increase our sample size, which is wonderful. And it makes it a lot more fun for us to review those survey data at another time. So some of the traits that we had taken a look at included attached or free air lobes, your ability to roll tongue, and also whether you have a hitchhiker's thumb, a widow's peak, and also color of your eyes. Now, there are actually other traits. For example, the way you would overlap, crisscross your leg, which leg do you put on top of the other is another sign. And also the way you would clasp your hand together. It's another one of these indications of dominant and recessive traits as well. So when we talk about all of these traits, one of the ones that is pretty obvious is skin complexion. But perhaps you already know that skin color can actually be in a very different form, known as albinism. When you look at these three pictures, one thing that should pop up in front of you right away is the amount of white color that you see. In essence, all three of these organisms are lacking in the production of melanin, which is a pigment that is necessary for including colors in your skin. So albinism is actually a congenital disorder. It is characterized by either a complete or partial absence of pigmentation in the skin. Some of the more noticeable areas include the hair and the eyes. Now, because of this lack of pigmentation, it can actually cause vision defects. Some of those include photophobia, where there is a lot of sensitivity to light. So individuals that have albinism tend to shy away from very, very bright light areas. Another one is nystagmus, which is repetitive, uncontrollable eye movement. So you see the eyes shifting to left and right repetitively, almost in an uncontrollable manner. Amblyopia, it's commonly known as lazy eye, where one eye fails to achieve visual acuity, so they have trouble seeing images in focus. And they also tend to have skin sensitivity because they don't have much melanin on their skin. They are much more susceptible to sunburn and skin cancers as well. Now, this type of albinism, it's not limited to humans. Other examples include rattlesnakes and also wallaby, although they do tend to happen naturally in very, very small numbers in nature. Here's another example of mutations that can cause drastic differences in the physique of a molecule or a particular structure. Take a look at this. You are all familiar with the characteristics of a normal red blood cell. Some of you may have already learned about the sickle cell trait. Now, if you look at the image for the sickle cell trait to your right, in photograph B, you will see some cells that are pretty normal and the other ones that are very oddly in shape. They look like the red blood cells has been stretched. That's called the sickle cell. And these are the ones that are around the background. So those are normal, but these are not, which indicates to you right away that this individual is a carrier for sickle cell anemia. So what exactly is it? So let's take a look at what forms a red blood cell. Hemoglobin molecule is found to be found and in the red blood cell itself. The hemoglobin molecule carries 4 different ions on it, which is necessary for carrying oxygen molecule. Normally, it is oval in shape. Each of the hemoglobin can bind onto 4 oxygen molecules, and a normal red blood cell can live for 120 days, right? That's about 4 months. However, if a person inherited a mutation, now this is actually one type of mutation that we'll take a closer look at later on when we get into molecular genetics. This type of mutation is called point mutation, also known as base substitution. And it tends to happen in reference to this particular disorder on autosomes. So these are mutations that happen on regular body chromosomes, not on the X or the Y chromosomes, which would then affect only one of the sexes. But in this particular case, the point mutation occurs where there is a single base within the genetic sequence on the chromosome that was changed. And because of that one single change, and what I'm referring to are the alphabets that you're probably familiar with, the nitrogen bases, adenosine, cytosine, guanine, and thymine, A, C, T, and G. It's one of these bases that are changed, which results in the sickle cell trait. This particular condition affects a lot of people. Approximately 1 to 3 million Americans have it. Of those, 8 to 10% of them are African Americans. Now, the problem that we're having is that when the hemoglobin and the shape of the red blood cell changes, this red blood cell that has a sickle shape can only live for about 10 to 20 days, as opposed to 120 days in a normal red blood cell. Some of the symptoms that these individuals would have include anemia, which is a lack of oxygen. They tend to have pain crises, which means that the red blood cells have a tendency to easily create a blockage in a blood vessel. So if you look at this particular diagram where my cursor is pointing, right, they tend to clutter, they tend to clog inside the blood vessel and creating a blockage. And that can also lead to swelling of hands and feet, frequent infections, delayed in growth or puberty, vision problems that can lead to damage in retina. And the dramatic combination of all of these different symptoms actually will shorten a person's life expectancy. Most of these individuals have a life expectancy of 42 to 47 years old. Take a look at this particular image. Now, if you have normal eye vision, it would be a piece of cake for you. You would probably notice right away, most of the background, it's comprised of dark green colored dots. And then what pops up would be the number 96. However, if you have a color blindness, which is, by the way, a sex-linked trait, a mutation that can occur on one of the sex chromosomes, which can lead to color blindness. So this particular condition affects more of one sex than the other. In majority of the cases, color blindness, because the gene that controls the ability to see color is located on the X chromosome, females need 2 defective copies before they exhibit color blindness. However, for a male, because you only have 1 X chromosome, depending upon whether you have the normal gene, which means you can see this color right away and with no problem, you can tell me it's number 96, so you don't have it, congratulations. But if you have trouble looking at this particular image that I had just projected, then you have a defective gene on the X chromosome, such that leads to color blindness. So who was the first individual to document the presence of dominant and recessive traits? Many of you have heard of his name. It's Gregor Mendel. Gregor Mendel lived between the years of 1822 to 1884. He was an Austrian monk, and what he was known for was the study of pea plants that was quite prevalent around his monastery. And these pea plants were special. They're special because they have 7 very distinct traits. They have a particular seed shape that comes in 2 contrasting forms, a dominant form and a recessive form. So for seed color, there are 2 different ones, yellow versus green, smooth versus wrinkle. For pea pod shape, there is in the inflated shape versus constricted. For pod color, it could either be green or yellow. Flower color could either be purple or white. flower location could either be at the leaf junctions on the tips of the branches on the very top of a plant, or they tend to be really tall versus being really short. So what makes it really effective in performing the task that he was studying is that because these traits were so clear cut, it makes the calculations and the tracking of these traits much more easily. Now, have Mendel selected a different kind of plant where there was some type of mixture or range of colors or height, then the calculations would have been much more harder to detect and to figure out that there are natural laws that are governing how these genes actually operate. So the other thing that makes his study really fascinating is that because he was studying pea plants that were really fast growing, he can study many generations. On top of that, these are not plants that are particularly large, so that it is much easier for him to grow in a garden and also to have large numbers of them. So how did Gregor Mendel lay the foundation for modern genetics? He actually provided insight into the relationships among genes, alleles, and chromosomes, even though he actually did not use any of these words during his time. Because remember, the chromosomal structure of how it actually it's formed was not published until the mid-1900s. So there's going to be an article about Gregor Mendel's biography, which is a very short article that I'll be sharing with you. So you take a look at that at your own time at another time. But let's take a look at what he discovered. Gregor Mendel discovered the first law, which he called the law of segregation. So what does it say? What he inferred was that for any given trait, there are actually different versions of a gene called an allele. These alleles would be inherited and passed on to the next generation. For any given trait, for an organism, there would always be two alleles. You get one from each of your parents. Each of these alleles will then be segregated or separated during gamete production, during the process of meiosis, meaning that they move away from each other. They're not linked. They don't always have to stay together. Each of the gamete, because they are not linked to each other, each of the gamete will contain one copy of each of these genes that controls a particular trait. And so depending upon which one of these two copies of the gene, whether you got it from the copy that's coming from mom or copy from dad, it would result in an offspring with a different phenotype. Phenotype means the physical attributes of the offspring. So let's take a look at some of the terminologies. We say that an organism are homozygous parents when they have a trait that is controlled by two alleles that are the same. For example, you have dominant A and dominant A in this particular parental cell. So when this parental cell, it's going to undergo meiosis to produce its own sex cells, well, as you could expect, the only gene that they will pass on is the dominant A allele, because they only have that in their body cell. They don't have any other one for this particular trait. But a homozygous parent can also look like this, where they have two homozygous recessive alleles, meaning that they have both of the same, but both of these alleles tend to be recessive. So in the presence of a dominant allele, when you inherit one from each parent, as long as you have one capital A or dominant A, the dominant A will express itself, masking the expression of the recessive allele. All right, so now, again, if this particular cell were to undergo meiosis and produce sex cells, the only allele that this parent can put in would be the lowercase a. You would not expect this one to produce a dominant A because it never harbored it in the first place, all right? And the same is true for the cell on above. So what about heterozygous parents? Heterozygous means different. They have different allele combinations inside the zygote, which is where the term comes from. So this parent would have a capital A and a lowercase a, so a dominant allele and a recessive allele. So when this particular parent undergoes meiosis to produce its own gametes, it actually can produce two different sex cells that can offer the next generation with a different variation of that trait. So if this dominant allele codes for, say, brown eye colors, and the recessive trait codes for blue eye colors, well then, if the offspring in the next generation received this particular, say, sperm cell, then the baby would be definitely expressing the brown eye color gene. Because no matter if you have one dominant A or two dominant A, it would still result in brown eye color. But if you have one dominant A and a recessive A, the dominant A will mask the recessive A. Only if you get two recessive A alleles would the baby result in having blue eye colors. And by the way, by looking at combinations like this is also one of the reasons why sometimes people are quite surprised if they have if two parents have brown eyes, all of a sudden they produce a child with blue eyes. It could be that, and one way to explain is that both parents are actually heterozygous for their brown eye color trait, meaning that both of them probably have one dominant allele and one recessive allele for the eye color, and the baby just will happen to have received two lowercase a's, one from each parent for that particular eye color. I hope that example would help you. Okay, what about the second law that he proposed? He proposed another law called the law of independent assortment. So what he says is that the inherited patterns of one trait, whatever that one body trait may be, will not affect the inherited patterns of another one. So I could get one color, I could get a dominant allele for eye color, but I could get another gene that codes for being short. Just because I get a dominant allele for eye color doesn't mean I have to get a dominant allele for my body height. And I think you understand that. So when is this not true? And I think you guys probably know the answer. That's when genes are linked together, meaning the positioning of the two genes are very closely in line with each other when they're found on the same chromosome. Then you tend to inherit that set of combinations together. All right, we'll have more discussions about that later on. So let's take a look at this on the visual diagram, all right, independent assortment. So take a look at this particular column here. We have two pairs of homologous chromosomes. Again, each pair of chromosomes controls different body traits, but within this cell, you have the darker color from one parent, the lighter color of the same color from another parent, all right? And the same is true for this one and this one on the bottom. Now, if you take a look at this, each one of these pairs of genes controls a different trait. When they align themselves during metaphase 1, you know this is metaphase 1 because of the fact that they are homologous pairs, right? Now notice the darker colors on one side and the lighter colors are on the other side. And when they separate and they form two different sex cells at the end of telophase 1, two of the darker colors went to this side and two of the lighter color chromosomes went to the other side. Sorry about that. My son just ran in here. Okay, so as I was saying, the two darker colored chromosomes have aligned themselves at random to one side of this sex cell at the end of telophase 1, and the lighter colors went to the other side. Now, when you continue on meiosis 2, right, all of the lighter colored chromosomes on one side. So two of the sex cells out of the 4 will be the same. And if, let us just say, if the darker color represents the dominant allele and lighter colors represent recessive alleles, then whomever the offspring may be that would inherit these two sex cells, it would actually receive 2 copies of recessive alleles. And if the offspring, future offspring, winds up fertilizing an egg cell using one of these 2 sex cells, well then the offspring will then result in having dominant alleles for those 2 different kinds of traits, whatever they may be. Now, take a look at this second possible arrangement. Again, we're talking about independent assortment, meaning different traits can be inherited separate from each other without affecting each other. In this case, the alignment is a little bit different. I have one darker color chromosome here on the right, and another darker chromosome, blue color chromosome on the left. And then they go on to line themselves up in metaphase 1, and then they are separated in again later on in meiosis 2, and the resulting combination, because we have dark color, light color, dark color, light color mixed up together, you wound up with different combinations of alleles that are coming together that are not the same. So you can have a dominant allele and a recessive allele for 2 different traits, like I said earlier. Just because I have brown eye color doesn't mean I have to inherit a gene to say that I would be very tall, although that would be nice. So that's what independent assortment is referring to, that different traits, the way what you get for each trait does not affect the other one. Okay, now let's take a look at this. So how did Gregor Mendel figure all this out? Let's take a look at the pea flower that he was looking at. This is what an intact pea flower looks like. And on this particular pea flower, it actually has all of the stigma and the anther, which produces the sperm cell, and the ovule, which is on the bottom of the flower here in the ovary. This is what we call perfect flower. This particular flower can actually self-pollinate, which means the pollen, when it's ready, would be released over here. And actually, because this part of the stigma, it produces a nectar or a sticky substance, it actually, the pollens, with the help of wind or some other insects or birds, can land onto the stigma. And then the pollen, which is basically sperm cell, will digest its way down into the ovary, into this flower, causing self-pollination. However, if you imagine having multiple of these flowers next to each other and they're all in full bloom, where the reproductive parts are exposed, just like you see on this particular diagram to our right, they can cross-pollinate then. The pollen from one flower can then be blown onto the stigma of another flower. That's what we call by cross-pollination. Now, why does that matter? Let's take a look. So, what Gregor Mendel did was he took the pollen from a purple flower plant. He actually took a plain brush, and at the time he actually took some of the and one of these bee insects that has the fur on it, and dabbed it onto the stigma of the white flower plant, because he wanted to control what he was transferring, as opposed to letting wind do its random job and take it when wait for chance to happen. He cross-pollinated these two flowers, and then he watched and see what kind of flowers would be produced in the next generation. So we call these two original flowers, where we got the pollen from, the parental generation, and we designate it with a capital letter P. The offspring that the parental generation produces, we call it the first generation offspring, and we designate it as F1. The results of this experiment show that all of the flowers in the next generation turn out to be purple. So what does that imply? The purple color, which is designated, which is controlled by a particular genetic segment, it's actually more dominating, the purple color is more dominant than the white color. So now he went on to study this. He said, well then, if what I see is purple in this flower, what happened to all of the, supposedly the genetic factor that is in this white flower? Where did it go? Did it get disintegrated? Did it get hidden? What happened? Was it really totally lost? So then what he did was he took the first generation offsprings from the F1 and cross-pollen and self-pollinated itself. All right. So then what he got was resulting a 3-to-1 ratio. He repeated this set of experiment many, many times to verify that his results were reliable and that they were accurate. So, but every time he did it, he found that this was the ratio that he will end up getting. That 3 out of 4 of these plants would be purple, and a quarter of them would be white. So what does this white color imply? It implies, it verifies that the purple color is in fact a dominant allele, a dominant trait that would mask the expression of the recessive trait. Now, all of this scientific explanation the terminologies that I just used, like alleles and genes, that came much later. He wrote it in a very different way, but it's already clearly implied that he understands that something was working inside these plants that managed the expression of the trait and was being passed from one generation to the next. And the resulting offspring from the second generation, it's called F2. So how are single traits inherited? Well, for any given trait, it's a dominant trait and there are recessive traits. They are aligned on homologous chromosomes when we inherit them from our father and mother. So what about these terminologies that comes up in your reading? Genotype versus phenotype. You probably already figured this out. Genotype is the genetic combination of an organism. So brown eye color could be homozygous dominant, meaning it could be big letter B and big letter B, because you get one big letter B from each parent. It could also be homozygous recessive, which means you have little lowercase b and lowercase b, because you got one recessive allele from each parent. So in both cases are homozygous, but one is homozygous dominant, the other one is homozygous recessive. Well, what about phenotype? Phenotype refers to physical attributes, meaning the looks of the organism. Let's put this into an example. So in the original experiment that I had just shown you, what Gregor Mendel did, if we have to use the modern science to explain it, each one of the alleles are represented by these letters. Capital P represents purple color, lowercase p represents white color. Because every single trait is determined by two alleles, you get one allele from each parent. So this purple flower has it's homozygous dominant. So big P, big P. And when it produces the sex cells, it would have only dominant alleles inside of it because it doesn't harbor any recessive one. That's how we figure this out. Now, for the white flower, it would be represented by lowercase p, lowercase p. It is a homozygous recessive trait. And its own sex cells would only encase the lowercase p and the lowercase p, because it does not harbor any of the dominant p alleles. If it did, it would be a purple flower and not a white flower. Now, let's take a look at this. To explain how this all happens is that for the next generation, they could have inherited a sperm that has a dominant p allele and a recessive p allele. If you get that combination, then the offspring would have big P, little p. In this particular order, you will get a purple flower. Or the offspring could have inherited a recessive P allele and a dominant P allele from the egg cell and result in this combination. In reality, it really doesn't matter whether you get the dominant or recessive allele from the father or the mother in this case, the final expression in the phenotype will be the same. All right, now, this is what shows the genetic combinations in each one. Students, sometimes I find students writing down lowercase b and then capital P, and regardless of how you write it, the final phenotype would just be the same. But typically, in standard notation, we typically would write down the dominant allele first, followed by the recessive allele. So it would be better if you keep it that way. Now, for recessive, it's always going to be two lowercase alphabets that is used as a designation. So how about Punnett squares? How does this apply in Punnett squares? Well, Punnett square is a mathematical calculation method that you can use to make predictions of what would happen in the F1 or F2 generation or any other subsequent generation. So let's take a look at how it works. Punnett squares are used for making predictions of genotypes and phenotypes, right? Because if you know the gene combination and you know which one's dominant, which one is recessive, then you know what they would look like. Okay, so let's go over anyway. We typically assign a capital letter for a dominant allele, like for free or unattached earlobes, and a lowercase letter for attached earlobes or recessive traits. We determine all of the types of genetically different gametes that can be produced by the male and female parents. So for instance, if it's a purple color flower, would the purple color be a result of big P, capital P, or a lowercase p, or a capital P and a lowercase p, right? But if they're white flowers, then definitely it would be lowercase p. Why do we need to figure that out? Because after when we figure that out, we then label it into a square like this if we are dealing with a single trait. If we're dealing with more than one trait, then the Punnett square would not look the same. That's a challenge I will give you later on towards the end of this lecture. So when you draw this square, this Punnett square, how do you label it? You would put all of the possible genetic combinations or the genotypes of one parent on one side and the genetic combination of the other parent on the other side. Now, you see how here I would put down the, I actually swapped the two, I'm so sorry. This should have been the mother and this one should have been the father. But you get the idea. It doesn't matter which one of the two parents you label, as long as you keep all of the genotypes for one parent on each side. Now, in this particular case, this parent that is the mother, again, ignore this label, has two sex chromosomes, X and X. And this particular individual, which should be the father, again, is mislabeled here, it's X and Y. Based on the genes, the chromosomes that are located in each one of these two parents, what are the odds for me to have a daughter versus a son? Which is what I'm using this Punnett square for calculating. So if I inherit one X chromosome from the mother and one X chromosome from the father, then I would have X and X. Certainly that'll be a daughter. And the same is true for this particular combination. But what if I inherit an X chromosome from the mother, but a Y chromosome from the father? Then I would have XY. Well, if that was the case, then I have a chance of having a son. And then the same applies here. So what does this show? That means if you were to make a prediction, you would have 50% chance of having a daughter and 50% chance of having a boy. And by the way, you and I know that because the sex of both parents don't change genetically, every time when they choose to have another child because they're wishing for one sex over the other, they would always have a 50% chance of one or the other. They do not have any higher chance of having a child with one sex over the other just because they were wishing for one. And I told you funny stories about that, where some of my extended family members have tried to have kids of one sex or the other. They wound up with 4 that were all girls or 4 boys. Okay, so now based on this, we can use the fraction or the percentage of this to estimate the odds. Now, obviously, these are probabilities. They're not guarantees. So if you want to achieve this ideal outcome that is mathematically calculated, you would need to have a large sample size, right? Kind of like flipping a coin. If you flip it 2 times, it doesn't mean you're guaranteed to have one head and one tail. If you do it 100 times or then 1,000 times, then yes, you would approach the ideal percentage that you estimate. Okay, so now, in this particular case, going back to the flower color, so here is the purple color flower that has been self-fertilized, right? So the same flower would have appeared here. So dominant P allele, lowercase p allele, and these are the sex cells that the genes that it could put into each of the respective sex cells. And you should not put 2 letters in here, because if you fertilize it with another sex cell, then you wound up with 3 copies of the same genes, which is a mutation, right? That's why under normal circumstances, there would only be one of these 2 alleles at randomly distributed either between these 2 options. And the same is true for this over here, because we're doing self-fertilization. And if you take a look at the possible genetic combinations from the egg and the sperm, this one will become purple color, but it's homozygous dominant. This one is heterozygous dominant. Heterozygous because it has 1 dominant allele, 1 recessive allele, and we call it dominant because it's the dominant one that would be expressing itself. The same is true for this one here, even though the order for the alphabets are written differently, but the consequence is the same. And then the 2 recessive allele down here would result in a recessive trait. So that is how the Punnett square actually works. Now, this is the mathematical calculation. Essentially, that's represented by here. I have 1/2 of a chance of inheriting this particular XL, and I also have 1/2 of a chance of inheriting the other XL. Well, the same applies for each one of these 2 sperm cells. So what are the odds for me to inherit this XL with this sperm cell? It's 1/2 times 1/2, which becomes 1/4. And the same is true for each one of these phenotypes that I see here. So that's where the calculation comes from. That's the genotypic ratio for a monohybrid cross. Make sure you know that term. And the ratio that comes out, it's classical. It's always going to be 1 to 2 to 1. Now, the phenotypic ratio, meaning the looks of the individual, would be a little bit different. It would be a 3 to 1 ratio. About 3/4 of them would be purple colored, reflecting the dominant trait, and 1/4 of them would reflect the recessive trait. Okay, all right, that's interesting. But how can we figure out whether a dominant trait that we're looking at it's heterozygous or homozygous for that trait. So for instance, when you see this purple color flower, we know that there are two different possible combinations for it. So what genotype can result in the purple flower petals? Well, if you were thinking, well, it's got to be either big P, or big P, little p, then you are right. It's got to be one of these two. But how can you verify whether the purple color is a result of homozygous dominant or homozygous recessive? Or heterozygous dominant. So the solution is to conduct what we call a test cross. So essentially, you would use a pure recessive or homozygous recessive trait flower that has a recessive allele and see if you can get it to express itself. Because if one of these two purple flowers is heterozygous, that means it harbors a recessive allele right here. And then in the next generation, there would be a chance for the offspring to show the white flower trait. But if the purple color flower is homozygous dominant, no matter how many times you do a test cross between the unknown purple flower with a white flower, you would always result in purple flower, and you would not be able to get this white flower to come out if you do this experiment enough number of times. This is what we call a test cross. It's used to figure out the genotypes of a particular trait that the organism has. Okay, for your practice, what I want you to do is using what you have learned, either doing Punnett squares or by sound reasoning, help me figure out the genotype for each one of these individual family members. Figure out the ones that have a number next to them. So you have essentially 11. I erased one of them and I gave you the answer for some of the other ones so you can see. Now, this is what we call family pedigree. It's very interesting. The horizontal lines that you see up here represents that these two individuals have a sexual relationship. And in this case, it's between a female represented by the circle and the square that represents a male. These two individuals got together, they produce two children. How can you tell? The vertical lines that descend from the two of them, these are their kids. They produce one male and one female. The female, it's color-coded in red, just like the father did. That means they can tongue roll. Red color means they can tongue roll. And being able to tongue roll is a dominant trait. So that would give you a hint. This daughter then went on to marry this gentleman over here, all right, that comes from another family. And again, the horizontal line indicates that they are married. And then the two of them, number 6 and 7, went on and produced two children. Again, one male and one female. So I want you to use some of the hints I've given you here to try to figure out the family pedigree. Now, here is something that is more challenging. I do give you the answer for this